is some of the sorry, guys, it's just this threw me off. Some of the kind of language that we typically use in genetics. So we talk about genetics and what genetic testing options are. So what are your providers, your genetic counselors doing in clinical practice? And then what we're going to dive into is an overview of variant interpretation, because we always have seen that this is very useful and rewarding to families to understand what this language means that you often see on the testing reports. And we'll talk about the variants and what they mean. Next slide, please. And next one. So this is SCN2A. So this is all about SCN2A, yeah? And just to decipher what SCN2A means, it basically means sodium channel number 2 alpha. So Stacy and I typically tell our families in clinic that gene names are often very difficult. So this is something, it's a combination of letters and numbers, and those are often abbreviations. And what it comes down to is that SCN2A is a protein, and a protein for a sodium channel. Here's one of our cartoons where we just like to kind of distill this into a single kind of piece of knowledge. So SCN2A is a channel for sodium. It typically sits at what is called the axon initial segment. This is a brain cell, a neuron. And SCN2A, in a kind of, let's say, simple overview, you can think of this as a key protein at the decision-making center of our brain cells that decides whether a neuron fires or does not fire. Next slide, please. Yeah. If there's SCN2A, there's also SCN1A, SCN3A, and many other genes. So the SCNs is an entire gene family, yeah? And this is not something that you necessarily need to kind of know. It's just important for families to know there are other sodium channels, and this is a very big family of brain genes where many people are doing research on. Yeah, so we're involved in what is called a Center Without Walls. This is a big NIH project where we are trying to curate and measure many of these changes. And this is interesting for us because it's not just SCN2A, but we know that SCN1A causes epilepsy, SCN3A, and SCN8A. Next slide. And then I'm going to hand this over to you, Stacy, to kind of start with Genetics 101. And I'm going to mute myself for the next few minutes.
Yes, so often when you're seeing a genetics provider or you're looking at a genetics report, it's often starting at a very baseline high level. And it's definitely useful to kind of start at the beginning to orient ourselves and make sure that we have all the information that we need to understand our genetic reports. So starting at the beginning, our body is made of cells, and these cells all have genetic material in them. And we get this genetic material from our parents, from our mother, and from our father. And this genetic material is put in packages that look like this. And so when you kind of unwind, you're able to see, this is the classic structure of DNA, this kind of structure. And this is where all of the instructions for making our bodies and making our bodies function is held. And this is what it looks like when you actually look at these packages of DNA or chromosomes under the microscope, if you lay them all out. And this is where SCN2A lives. It lives on the second chromosome, and we have 23 pairs. So this is where SCN2A is when you're looking in this zoomed out kind of way. So we, starting with the human body, we talked about the cell, we have our DNA package, right? And so we talked about the chromosomes and how they contain all of our genes. And the job of our genes is to make proteins like SCN2A, for example. If you break this down even further, there are actually different parts of our genes, and there can be changes in a gene throughout the entire gene. We're going to talk about what those changes might be and how they may lead to disease later. But really, there's two different major parts of a gene. We have what's called an exon, and these are the pieces that actually make it into the protein that does some job in our body, like becoming a sodium channel or things like that. And we also have these sections called introns. And these parts of the gene are not ultimately included when we end up making the protein using the instructions in the gene. So when a lot of people are diagnosed with SCN2A from what we call whole exome sequencing, so that's looking at this area of the genes, these exons. So we've talked about, we have these packages of genetic material in chromosomes, and there are many different ways that we can test our genetic material for genetic conditions. So the kind of highest level is a test called a karyotype, and that's actually exactly that picture that I showed you of the chromosomes earlier. So you're looking at those chromosomes under a microscope, and it's seeing, are there any major chunks that are missing or extra? Then if you zoom in a little bit more, there's a test called chromosomal microarray, and that looks for smaller changes in the amount of DNA that we have. So again, smaller pieces of genetic material or instructions for making our bodies that are either missing or deleted. And then we move into looking at the finest level of detail. So we're not looking at the chromosome in a larger sense, we're looking at specific genes. And there are a few different ways that we can do this in clinical practice. So the first is what we call a gene panel, which many of you may be familiar with, as your children likely had these, or whole exome sequencing. And the idea is, with a panel, there are many different genes in our bodies, but when a genetic testing lab makes a panel, they put together a certain number of genes that are associated with a certain disease, like epilepsy or autism. And they look specifically at the spelling of that gene to see if there are any harmful changes in that gene that they think is causing disease in the person that they're testing. And then there's also another way that we look at our genes on this finer level of detail, and that's whole exome sequencing. And that's looking at all of our genes to see if we have any of these spelling changes that are associated with disease. So the idea is that it's more broad than a gene panel, but it's still looking for spelling changes that may be harmful to the function of a gene like SCN2A. And this is often how families with rare disease reach their diagnosis, because it's not something that physicians often think to test for, specifically looking at that one gene for rare diseases. So I've been talking about these genetic changes, and this is what this is a cartoon to kind of represent what I'm talking about. So we have our DNA, and it's encoding instructions to make a protein like SCN2A. It does this using 4 different letters, A, T, G, and C. And the body reads this code and assigns a certain building block of a protein. So you can see there are different building blocks of proteins called amino acids, and each set of 3 letters encodes one single piece of a protein. So for example, if this is the way that a normal string of a protein is supposed to look, if there's a change in the spelling of the DNA, that tells the body to put a different piece of a protein in that spot. And sometimes when this happens, it makes the protein function in a way where the gene isn't able to do its job, or it does its job improperly. And this is what causes disease from a genetic mutation. So we have this spelling change here. We have this different piece of the protein than what we would expect. And the labs are actually able to name these changes, and that's what you see on your genetic report. So there are 3 different ways that we name these changes. The first is using what we call genomic coordinates. These are not typically used on testing reports. It's more on the genetics side and not so much important in a testing report. these are the two things that you're going to see on our report. This is called what we call C dot and P dot. So C dot is talking about the coding. So this alphabet here, it's saying, for example, on the 2,558th letter, there's supposed to be a G, but there's an A. And that's how they name the genetic changes, and they tell us where the change is in the gene. And then the P dot, tells us what happens in the protein as a result of this spelling change. So there's supposed to be a certain building block at this spot. There's supposed to be this building block. And this is, again, just the string of the protein that tells us where in the protein it is. And this is the amino acid that's there, or the building block of the protein that's there, that is different than what we would expect and may be associated with disease. So I want to thank everyone for getting their genetic testing reports to us so quickly. We very much appreciate that. I pulled up one genetic report that we received, and we're going to go over a lot more later. But I just pulled up this one as an example of what this might look like. So you see here the name of the gene, SCN2A, the disease associated, the mode of inheritance, which means how many mutations you need in that gene to have that disease. And then this is how they describe the variant in the same way that we talked about on the last slide. So when we think about looking at what a genetic testing report includes, at least on the first page, this variant, the way they label this tells us what's happening in the gene that we think is causing the disease, where we think it is, and what it's doing. And we will be going over a lot more examples later, but I just wanted to provide one example here. And I will turn things back over to Ingo for the slide.
Thank you, Stacy. So this is basically a version of the SCN2A channel that we had as a cartoon a few years ago. And this just kind of shows you where the mutations on the SCN2A protein are. So you have to imagine that we take the ion channel, which is typically a kind of round protein, and just iron this out and flatten this out. And the reason why I use this cartoon still is because it kind of shows that there are many changes in SCN2A, and some of them actually cluster. Some of them are in specific regions, and we know that those regions are very important functionally. And what you can also see is that there are different changes in the SCN2A gene that we have kind of annotated by different colors. They just refer to different kind of clinical presentations. Next slide. So this is now a more complete version. This is from our publication earlier this year from Katie Crawford and Julie Shen. And this maps all the existing, or all the reported changes in SCN2A on this protein. So what you can see at the top is, again, this flattened version of the SCN2A channel. And at the bottom, we've basically just made this a little bit smaller and just collapsed this, how this looks on the actual gene. Yeah. And what you can see here is that there are some changes that kind of stand out. Yeah, you can see these kind of higher changes, and maybe Stacy, you can just kind of hover over the 853Q or the R188Q, and those are changes that are seen in many children. Yeah, and we call them recurrent variants. And this is one important thing when we talk about genetic changes, how we interpret them. This change has been seen previously in a child with SCN2A. Next slide, please.
Okay,
so back to you now, Stacy, right? Yeah.
Yes. So Ingo, Dr. Helbig has nicely talked you through where these changes are. So now we'll talk a little bit about what these changes are. And there are many different types of genetic changes that can occur. And we're going to go through each of them and their context for SCN2A specifically. So The first type of genetic change that we'll talk about is what we call a missense variant. So this is what we've touched on earlier, where you're supposed to have this CAT spelling here, but instead of CAT in the genetic code, you have CCT. And so you change the corresponding building block of the protein, you change the protein, and you change the way the protein works. And this is the most common type of disease-causing variant for people who are affected with SCN2A. About 75% of the time, this is what we find when we test people for this gene. And they have this disorder. And this is what it looks like on a genetic report. So you have the C dot, and again, that's corresponding to this spelling here. It's telling you it's on the 2558th letter in the spelling code. You're supposed to have a G in a certain spot, but you have an A instead. And this is what this means for the protein. So again, this change in the code makes the protein different and then causes disease. That's the idea with a missense variant. We also have nonsense variants that can be seen with SCN2A. These are less common, so about 5% of people with SCN2A related disorders have one of these changes. And again, so we have our DNA code. It's telling us how to make the SCN2A protein. And the protein is supposed to be a certain size. But some of the genetic code actually tells the body to stop the protein. So even if there's supposed to be, let's say, 100 more pieces of the protein after, it stops too early. And so you're not making the complete protein. And again, it's not able to do its job properly. So the way that we indicate this on what we call the C dot is the same as with a missense variant. But what if it's coding for a stop signal, it makes the protein stop. And this is what it looks like when we write this out in our genetics nomenclature. So this little star here just means that at this position, the 100-second amino acid or building block of the protein stops. It stops too early before it's supposed to. And so it doesn't make a complete protein. And this is a frame shift variant. So this is a little bit more common than what we've talked about on the last slide. About 8% of people with SCN2A have what we call a frame shift variant. And so another type of change that we can have in the genetic code is actually adding an extra letter in there that's not supposed to be there. And you can imagine that you're supposed to have CAT here. You have ACA, it throws off the rest of the protein. And so all the rest of the code after this is inserted, you get all the different amino acids afterwards. And so what does this actually mean? It means that you have the SCN2A protein with incorrect amino acids, and it may stop early. So that's the idea with frame shift specifically. So when you're adding in these incorrect amino acids, sometimes it codes for one of these stop signals again. So again, you're making some segment with incorrect protein, and then you're stopping the protein again too early. And that's the idea of how this causes disease. Next, we have the splice site variants, which is a different type of genetic variant. This goes back to what we were talking about a little bit earlier, these two different pieces of what a gene includes. So the exons that make the protein, and then the introns that are between them. And one of the things that our body does is cuts out the intron because it doesn't need it when it's making the SCN2A protein. What a splice site variant does, you can see what this looks like on a report here. What it does is it changes the way the body puts the exons together. And again, the exons are those coding regions that actually make the protein. So let's say you're supposed to have 1, 2, 3, but these splice site variants change the way that these exons are put together. And so we're skipping exon 2. So we're skipping an important part of the protein that is needed for it to function. And that is how it causes disease. And so whenever we're talking about some genetic change in an intron, this is what it looks like on a report. This plus sign and a 1 means that it's the first nucleotide into the intron. It's the first letter into the intron. And that's very important for telling the body to take that intron out. And so if there's a change there, it's not able to do that effectively. All right, and then on a different type of genetic change that can cause SCN2A are what we talked about earlier. So this idea of having extra or missing pieces of DNA. So really thinking about, we know that SCN2A is on chromosome 2, and sometimes people have segments of chromosome 2 that are missing or extra. And this is, again, a less common way that people can have SCN2A related disorders. About 6% of people have SCN2A related disorders because of this change. So you can imagine that having the gene missing, obviously you're not able to make enough of the protein. And that is how it causes disease. All right. And then, so we've talked so far about SCN2A as a protein. We talked about the different types of changes that can cause disease when someone has changes in this gene. But we all have genetic changes, right? That just make us different people. And so what the job of a genetic testing laboratory to do is to collect evidence to see if they think that a change is disease causing, or if it's a change that any healthy person can have and it doesn't affect their health. So how do they do that? Luckily, they actually have a standard set of guidelines that are put out by the American College of Medical Genetics for exactly what types of evidence and how strongly to weight the evidence that they have in order to make a decision about whether they think a variant is disease causing. So these are the so-called ACMG guidelines. So the American College of Medical Genetics has these guidelines, and the idea is we want all laboratories to use the same types of evidence and weight the same types of evidence the same way when they're making these decisions to make it a standardized process. So these guidelines provide standard terminology for how to classify a variant in these 5 different ways. We're going to talk about them a little bit later, but pathogenic and likely pathogenic are words for things that we think are likely someone's genetic diagnosis or what's causing their medical symptoms. Uncertain significance is a little bit more complicated. Sometimes it is someone's genetic answer, sometimes it's not. It often means that the lab does not have enough evidence to know for sure whether it's just a normal change that anyone can have, or if it's a change in a gene that can actually cause disease. And then likely benign and benign are changes that are not reported when you actually look at your report, because it means that they don't have any implications for someone's health. So this is the idea. They have this 5-tier terminology that all labs should be using. And I just wanted to show you this little table here of different types of evidence that they look at. This is not something that we'll go through in detail, but they're supposed to use these kinds of criteria, these types of evidence. It's very, these criteria are standard and all labs have access to these, and they use these and weight these hypothetically in the same way. So there were a lot of words on the last slide, but really there's just a few concepts that are important to think about in variant interpretation or thinking about whether a change in a gene is disease causing. So one is population frequency. So this means, do we see this genetic change in cohorts of healthy people? And for conditions like SCN2A, we would not expect to find harmful genetic changes in healthy people. So if someone comes back with a genetic variant in SCN2A and it's been seen 1,000 times in healthy people, the lab says, okay, this is likely not someone's genetic answer. There's what we call in silico prediction, which are just computer models that tell them whether they think that this is harmful to the gene or not. There are functional studies. So you can put a genetic change in a cell and study it in a lab to see how it affects the way that the SCN2A channel works. Phenotype. So does the person that you're doing genetic testing on fit the phenotype, or what we think of as the picture of that genetic disorder? So, so things like epilepsy, autism, things like that. Segregation data is a big piece of this as well. What this means is testing parents. So as many of you, I'm sure, have been through this process, often it's helpful to know if a genetic change in a child is brand new in them, or if it's something that they got from a healthy parent. And if it's inherited from a healthy parent, it's sort of like having it in the healthy population. And so we usually expect that these conditions are brand new in a child. And then, as Ingo mentioned, there are recurrent variants in SCN2A. So have we seen this change before in someone who has SCN2A related disorders? And again, that's a piece of evidence that is weighted in variant interpretation. And now I will turn things back over to Ingo.
Yeah. And so, Stacy, would you be okay with a last minute change? Because I realized that we're already kind of 30 minutes in. And what I did while you kind of did your presentation, I copied all the testing reports. So we might actually jump right into this. Yeah. So what I'm going to do quickly, I'm going to share my screen. Mm-hmm. And I just would like go through a few tests. So we asked the foundation to kind of provide us with a few tests where the names are taken off. Yeah. So sometimes there may be a first name in there, but this is basically what we think of de-identified. And I just wanted to kind of go through some of these tests and first of all show you the different kind of formats how these tests can come back, and then have an idea or provide an idea on how these different tests actually tell us about SCN2A. So we're going to start with this change here, and I'm not going to put some presentation mode because I'm going to zoom in and zoom out. But this is now a test from a so-called SNP array. So this is a change that looks at little gains and losses of chromosomal DNA. And we can see this is a deletion that includes the SCN2A gene. So this is a genetic change that is disease-causing because it removes the entire SCN2A gene. This is only one copy of SCN2A. We're going to look at this next test. I'm going to go through this in a little bit of kind of a faster pace because we have lots of time for question afterwards. Yeah, we don't want to make this too formal. Here is a different change. This kind of change here, you can see what Stacy has told you about this what we call C dot P dot. So this is where the protein position is, the Y1771H. So this is the address of the protein change, and the C dot is the kind of genomic or the RNA or cDNA that kind of codes for this. And here you can say this is it's cut off a little, but it says pathogenic variant. And this is a variant that is de novo. So de novo means this variant is new in the child and absent in the parent. And this variant is kind of classified as pathogenic because it has been seen before. So this is a change that has been previously seen. I'm going to zoom in a little bit and you can see this change here, P dot Y1771. Let's look at the next test. So this, and you can see these tests come in many different formats. So when I put this together, we really have an entire list here of different changes or different testing reports. So this now we've seen some changes that are definitely disease causing. And now we have something here that is kind of something that is a bit unusual. So we have a change in the SCN2A gene. And we have here again the C dot and the P dot. And it's not a change that we've seen previously. And this change is what is called homozygous. So this individual here has two changes in the same position. And this makes this uncertain significant. So this is something where more work needs to be done, or we would not be able to kind of really put this into a clear category and saying this is disease causing or not. Here we need more information on, is this change new or is one of these changes new? And then kind of move from there and understand what we have. Here's our next test. And you can see not all testing reports are straightforward. So some testing reports are, they give us a very, very long list here. And you can see here there's actually father, mother, and the paternal grandmother. And we're trying to understand what is going on here in SCN2A. And we know that there's some symptomatic. It means that the grandmother may be disease related, but this is also a kind of SCN2A change from the previous test where we're not quite sure what this actually means at this point. This is not something we can easily say this is disease causing or not. Let's move on to this different testing format. And if this is blurry on your screen, it's also blurry on mine. And it's just one common problem that we have, that we sometimes have families and they have a genetic testing. They have the very expensive genetic test done. And sometimes these kind of test reports are traded down or are copied, or even that the lab scans this in. Then we have to say, well, can we really kind of see this here? So let me see what I tell you what I see here. So this is a change. This means de novo. And if this is a de novo change, it means that this change is new in the child. This is a change in SCN2A. And typically changes in SCN2A that are de novo, they are typically likely pathogenic or pathogenic. So we're quite convinced that there's a new change in the SCN2A gene. This is more likely than not the answer for a individual's epilepsy or developmental issue. So here, this is a test. We can see this is now in Spanish, but we can basically piece this together. We can say this is the genetic change. Here you have the G dot, C dot, P dot. So this is always the same. And this is why I'm showing you this, because we always look at the very same things. This is the position. We know that this change is present in the child, absent in mom, absent in dad. This is formally classified as a uncertain significance. And what Stacy and I would do in these situations, we would kind of look at what the child has and kind of understand that there's the genetic testing report. And then there's what we kind of make of this entire story. So we would call this a variant of uncertain significance. And then this would be explanatory in the context of the child's disease. And what is important here is that the genetic testing doesn't define what a child has. Genetic testing always needs to be interpreted in the clinical context. This is sometimes easy if the change is very clear. If this is a stop change or the entire gene is missing. But many of the changes we see are uncertain. And then it's really important to kind of say, this is the testing report, but then above this is the interpretation of your genetic counselor or your physician. Here's another change that makes this actually relatively straightforward. You can see here, I'm going to zoom in again, SCN2A. what you can see here is now the C dot. And what you've previously seen is that the C dot is usually a C dot A or T, a bunch of numbers, and then a different letter. And here you can see it's del CTTT. And here you can see the P dot also looks a little bit strange. Yeah, this looks P dot TRH 369, and then there's a long letter here. And what this is, it's a so-called frame shift variant. It's one of these frame shift variants that Stacy was talking about. This variant basically puts a frame shift into the SCN2A protein, and many of the proteins, many of the amino acids that have been produced are out of sequence, and this often leads to a stop. Yeah, these variants are always likely pathogenic or pathogenic. This is a loss of function variant, yeah? Which is we typically see with developmental differences, autism. Yeah. Let me look at our next change here. So again, this is the same change here. You can see the C dot with this del. Yeah, so it means that there is a deletion of 4 base pairs, and our genetics always kind of comes in 3. So we have 3 base pairs for 1 amino acid. If you have 4, you get a frame shift. Yeah. And I'm going to do maybe a few more because then I want to make this a bit interactive. So here we have a similar situation. We can see this is a C dot, and you can see the del here. Del means that this leads to an X, and X means that the protein is kind of stopped. These changes are protein truncating variants. Those are the same as stop mutations, yeah? And this leads to a loss of function. And then let me do one last here. So this is a C dot and P dot, and you can see the star here. Yeah? And if you remember what kind of Stacy was showing us earlier, the star means that this is a stop. So this genetic change introduces a stop at position 243, and this is a stop mutation and also a loss of function variant, because this introduces this kind of halt in the SCN2A protein. Here, we have some languages that's a little bit different, and this may sometimes be older testing or testing that is not done in the US, where labs talk about positive testing or abnormal testing, yeah, and don't use this kind of 5-tier system. So this doesn't give us pathogenic or likely pathogenic, but other kind of countries or other systems use other frameworks. But let's kind of try to kind of piece this together. This is a de novo variant. So it's a de novo variant that is new in the chart. Here they actually use the language pathogenic, yeah, and it's a missense change. It's a spelling change. Here is the C dot, here's the P dot, and this is at position 195, yeah. And when you can ask the question, how can the lab be so certain that there's a kind of change at 195 that kind of makes this causative? It's because other patients have been reported with this change. Yeah, so this is kind of pathogenic because it is a previously seen change. Let me maybe kind of take the temperature quickly, or just kind of get a bit of the feedback. Would you like us to kind of continue with going through some of these changes, or do we want to kind of do a round of questions and discussions? Jenny, what's the best thing?
I haven't seen any questions come in yet on the chat. Just remind everybody to check the chat for, you can introduce yourself and also to ask questions. Yeah, so I'm going to continue with a few changes then. And if you just let me know, just interrupt me or kind of change us or just kind of stop us quickly when you have questions. Okay. This is not meant to be a lecture. This is something where you can say, wait a second, I don't understand this. Yeah. So we've done a few now as practice. So please kind of interrupt us from now on, just to make sure that we can answer some of these questions that you have. Again, our kind of goal is to educate families on how to read a genetic testing report as the first step when it comes to knowing about SCN2A. Yeah. So type your questions in the chat or raise your hand, and I will interrupt for you. And if you mind just kind of monitoring the chat for us, because I'm sharing now, so this is hard for me to see. So please jump right in when there's a question coming. Sounds good. So I'm going to
do this a little bit slower now, waiting for kind of questions and comments. And let's look at this next test. Yeah, so here we have an entire bunch of genetic changes. Yeah, so this is typically what families often see when the first round of genetic testing has been done. This has been done on a so-called gene panel. And you can see that there are many genes and many epilepsy-related genes, or changes that this individual has. It doesn't mean that all these changes are causative. It just means that many changes that we carry are just kind of private to us, and are changes that need to be kind of worked up on whether they're kind of causative or not. Here's an SCN2A change, and you can see this kind of C dot here, again, looks a little bit different like we've seen this previously. So this is not a change from A to C or C to T or T to A. This is what it called intronic. Yeah, and you remember Stacy's slide previously that our genes are broken up into little blocks that are called exons, and then they kind of spliced together. Intronic means here that this is not within the part of the gene that codes for the SCN2A gene, but someone right next to it that may is part of the intron right next to it. And you may ask the question, why is this even on there? And this is on there because the question is this a splice variant? Is this something that kind of impairs the kind of stitching together of exons and may potentially lead to a missplicing of the SCN2A gene, and therefore is almost like a loss of function variant?
I do have a question
Yes.
in Ingo, and it's a good one. So if you've had a genetic test report done with the field changing so rapidly, how often should you ask for it to be redone? Is 2 years too soon, or is that a good number? I just had my son's redone because it was done in 2012. And it was a variant of unknown significance, and it is now likely pathogenic because.
Yeah. So I think the good thing is that you've already introduced this question very well. I think this is usually a question if there's some kind of lack of answer. Yeah, and in the SCN2A families, this would be a variance of uncertain significance. Yeah, what we recommend families is this kind of check in every year. It is often not the need to kind of redo the test. It just might be that there's more information for one specific change. Yeah, and the other thing that's important is there's not a test that needs to be, depending on what test was done in the beginning. We often don't need to redo the entire test. We don't need to give blood. Yeah, we can often just reanalyze or reinterpret the information that's there. If there's some uncertainty about this change, every 1 or 2 years, yeah, because this is where there's lots of information coming together. If a family or an individual has an SCN2A diagnosis, so if your provider kind of has done all the kind of work and say this is really a clear SCN2A disease, this is less urgent.
Right.
So it's very uncommon for changes to kind of go from pathogenic to not disease causing, because the framework that this is interpreted by is quite robust.
Right. So it's really if you don't have an answer.
If you don't have an answer, if you have an SCN2A change that is uncertain, yeah, every 1 or 2 years.
Yeah, that's great. Another question. Let's see. Are there certain mutations? Does the location of the mutation have any bearing on the symptoms?
Yeah.
And the question was particularly for gain of function, but
yeah. Stacy, do you want to kind of take this first?
I'm sorry, I was reading the chat and I missed your question.
Then I'll just answer this question. The answer is absolutely yes. Yeah. So can you just introduce the kind of word gain of function and loss of function? So when we think about SCN2A, on what a mutation in SCN2A actually does to a protein, yeah, we kind of think of this may decrease the function or may increase the function of the SCN2A protein. When you think about, and both can lead to these different SCN2A phenotypes, and the extremes are developmental problems, autism for a loss of function variant, and early onset epilepsy for a gain of function variant, with many types in between. What is happening with the protein is that typically, if you have a stop mutation, this can be anywhere. This can be very early, this can be in the middle, this can be outside where the protein passes through the membrane or not, so this doesn't really matter. If there's a gain of function change, a change needs to kind of be in specific parts of the protein to allow for gain of function. And typically the changes we see that result in gain of function, they're usually in very specific parts of the protein. And let me just kind of, Stacy, let me kind of pull up my presentation here. Oops, sorry. Where is our protein? So the way that you can imagine this, if this is our SCN2A protein here, yeah, in order to make this a gain of function. So think about this really as a channel where kind of sodium flows through, and gain of function means the channel is just more open. So you need to have a change that is here somewhere or sits there. Yeah. And when we look at the protein model, those are changes that typically sit in this part here. So this is the protein again ironed out. This is called a membrane, and this is the transmembrane changes. So for gain of function variant, it matters absolutely. For some loss of function as well, but you can have loss of function at any point in the SCN2A protein.
Okay, Omar had a question. Omar, would you be comfortable just asking your question out loud? You can unmute.
Hi, how are you? Can you hear me well?
Yes.
We can hear you well.
Yeah,
Thank you.
sorry. Sorry for my English. I am from Chile, so this is not my first language. But I just want to know. I mean, maybe I'm just thinking. But before that, thank you for the presentation. I think it has been a really clear presentation, and for us as a parent that we were not, I mean, used to see that kind of information, but it's just, I mean, I have seen what you were presenting, and I was just reviewing the intronic and the other information that you were mentioning. My question is, can a gene change after birth? I mean, that is what I trying to understand more a little bit, because what I see is that, I mean, the letters that you were showing us, it was a change between the letters, right, in the gene, but in the at the protein level. But I'm thinking if this can, I mean, as we have seen as a parent, that it was a change during the process. I mean, some of us see, I mean, the birth was a normal birth, and then we see the change after, I mean, a few months or a few years after the birth. This is something that could be changed during the process, or was something that was there, and then, I mean, we see what's the reaction of the issue after. But it's just a question. I'm not sure if it's a dumb question, but yeah. Thank you for your support there.
No dumb questions.
Do you want to take this question?
Yes, so certainly not a dumb question. And when we think about our genetic changes, these are things that we're born with and they don't change after birth. I think a lot of people are excited about the idea of what we call gene therapy, the idea of changing our genes to cure disease. But thinking about our kind of natural biological processes outside of this, we are born with changes in our DNA. We all are, not just people with genetic diseases that are different from our parents. We have these genetic changes, we're born with them, and we have them throughout our life. Our genetics don't change in that way. Yeah. Not a dumb question at all, though.
Yeah, I think this is a very good question. I would agree with kind of Stacy. It doesn't actually, sometimes the symptoms of SCN2A may only be apparent later on, yeah, but the change has been there from the very beginning. So those are changes in that a child had from the very early stages during pregnancy even. Yeah?
Thank you so much.
Any other questions right now, or should we?
Christine, you raised your hand.
I did.
Yeah.
My question is there any research right now that would suggest certain variants are more likely to shorten life expectancy?
So this is a, let's say, broader question. I would like to give you maybe the answer that I would kind of give generally. So when we think about life expectancy in children with SCN2A related disorders, it's not so much the genetic change, but it is the severity of the individual's disease. So we know if someone has very severe epilepsy, and has many symptoms that a child cannot walk or talk, then we know that these symptoms may contribute to that the child is at risk for medical issues that may eventually be dangerous, or may eventually lead to changes that could endanger this child's life. But it's not the genetic change per se. But when we think about how we can break this down for SCN2A, we know that both loss of function and gain of function changes can result in very severe but also very mild epilepsies. And it's mainly in the children with epilepsy where we're concerned about ongoing seizures and seizures that cannot be controlled, but we have to talk to families about how we can minimize the risk.
Thank you.
Ricardo, you had your hand up.
Hello, it's Grace from Brazil. How are you? And I have a question regarding redone the test. Do you believe that is important around 2 years later for the first test to do another one? What do you think about it? I don't know if you understand. I can repeat.
Yeah. So I must say I couldn't really understand. Maybe Jenny or
I think she was asking if it's important to repeat the test after 2 years, which we talked about a little bit earlier, but you might not have been able to hear what you were saying, Ricardo. Do you want? The answer is yes. If it's a variant of unknown significance, if you don't have a definitive answer, it would be a good idea to talk to your doctors about not necessarily redoing the test, but reanalyzing the test.
Yeah. So and I think this kind of links well to some of the previous questions. So our genetics doesn't change. What changes is our understanding of what changes mean. So many of the changes we have in our genetics in SCN2A, they might be uncertain, but we have neither, and what uncertain means is that we don't have evidence to say this is a benign change, but we also don't have evidence to say this is a causative change. And this information varies, and this information typically changes within a year or in 2 years. So if an information about an SCN2A change isn't clear, then you would look at this again after 1 or 2 years. Sometimes it's actually easier to redo a test, because a test comes with an interpretation, but this really depends on what tests we can do, where you live, how the healthcare system is. But the general idea is if the SCN2A change is uncertain, we would like to redo this again after, or we look at this again after 1 or 2 years. I just took my screenshot up so I can kind of see the entire kind of audience. Yeah.
That's great. Amanda, do you feel comfortable asking your question, or do you want me to just read it?
There we go. Thank you so much for this. It's great, and it doesn't feel as lonely seeing all your faces. We are in Maine, and we have a very, very good neurologist. We have access to very, very good developmental pediatricians, but our son's SCN2A, which I believe is a loss of function He has an autism diagnosis and a developmental delay. They aren't well versed in SCN2A, basically just gave us some paperwork and said, hey, that you could join. And so at this point, I feel like I'm more of an expert than they are.
Yeah.
Would you recommend, Dr. Helbig, a second opinion with an expert like yourself so we can have a better understanding of the plan ahead, other than just sort of right now, our doctors are just sort of looking at our son like, oh, it's just autism. And I want to be a little bit more prepared as far as things that we can do for him or to expect.
Yeah. So and maybe Amanda, if you don't mind, I'll just basically put this in a broader context. So basically, we have it is very common that we have very good pediatrician and neurologists in the community, but SCN2A, as you all know, is rare. Yeah. So this is something where while many doctors already know about this, knowing all the kind of the ins and outs and how we would interpret this is something that we would not expect a doctor in the community to necessarily know. Yeah, what has actually kind of changed over the last year with the kind of entire pandemic is that we've already been online all the time, and there are many programs that actually have telemedicine ways of doing this. So you might actually be able to kind of connect with one of the epilepsy genetics or neurogenetic programs virtually. Yeah, and what I also kind of often tell families is that this is not really kind of pitching your doctors against a different center. Many physicians are often quite happy if they kind of say, I get this additional expertise. Yeah, in many cases, it just kind of reassures them that they're doing the right thing. So I would definitely, if you have a question like this, non-urgently, I would say connect with any of the neurogenetics or epilepsy genetics programs. Yeah, and this is usually a very kind of rewarding experience for the family. Yeah.
Thank you so much.
Yeah, and I think we have, Stacy, maybe you can kind of share your screen in the end. You can have our contact details there, yeah, where it would definitely make sense to kind of look at this. Carla, you've been kind of patiently waiting with your hand raised. Do you want to kind of quickly ask your question?
Sure. There was a question in the chat, so I just wanted to make sure that they understood it. So maybe it was in regards to one of the genetic reports that you pulled up. It said the report that you pulled up that said that might have been done at another country, and it was done in the US less than 2 years ago by GeneDx. Should they request that it be updated?
So I think that this may have been the report that was positive. Yeah. If this was a clear, at least from what I could see there, this was a clear disease-causing change. Sometimes the language that has evolved over time. So sometimes when we look at the genetic testing report that we do, I think the labs have gotten a bit more kind of careful how they phrase this. I think what I stumbled across is that they kind of called a test positive, yeah, because this is typically not the lab's decision to make this, but it's basically the physician's interpretation. Yeah, so the lab can only give us an interpretation, but then the final diagnosis is kind of made by the person who goes through this test with you. Yeah, but from what I can tell you, there's nothing that I say this is out of the ordinary. It's just the language was a little bit different.
Okay, thank you.
Christine, did you have another question? No? Okay, your hand was still up. It's okay. Okay. Any other questions before we move on? All right, I think we're at a lull in questions. If there's something else you want to bring up.
There were a few other slides we had, although I don't want to oversaturate everyone's minds because I know we went through a lot of technical information.
I have a few more tests. So if no one has questions, I'm just going to fire off some testing reports here, okay? Let me just share my screen again. Yeah. And so this is the test I kind of showed you previously. This is the test that has these different changes. Now let's look at the next test. Yeah, that is basically, again, there you can see this as a test that was done in the UK. So here we have a different kind of framework on how this is reported. Yeah, where this change is mentioned here, and we can see that this change is at the position 880. Yeah, and what it says here is, you can now almost follow along. It says this variant has not been reported previously. Yeah, and in addition, this is new in the child. So the parents did not carry this mutation. So this is a de novo change. Yeah. So this is a change where we can say this is a change where we know this is a part of the protein that has never been seen in a different mutation. The lab report would probably kind of classify this either as a variant of uncertain significance or as a kind of likely pathogenic variant. And what I typically tell families in these cases is don't be too concerned about what the final classification is. So this is something that really needs an interpretation in the disease context. And we would probably kind of say, even though this may be uncertain or maybe likely pathogenic, we would name this disease causing in the context of what the individual has. Yeah. So here is our next one. Yeah, and you can see there's basically an entire range of what these reports look like. So they come in all colors, on all font sizes, table like this, table like this. Here we have the following, yeah. SCN2A, and you already know this is the C dot, yeah, and here's the P dot. And typically what labs do, and it's actually very important to talk about this quickly. The labs often put some kind of language on here that says associated disease. This doesn't say anything about the individual who had this test. Yeah, this is often sometimes confusing that families come back and say, why does it say epilepsy there? My child never had a seizure. Is my child going to have seizures? No? And the answer here is actually very simple. This is just the kind of, they looked this up in a dictionary, and this is what SCN2A, in this case, is called in this database. This doesn't have anything to do why this test was sent. This doesn't mean that this child has an early infantile epileptic encephalopathy 11. Yeah, it just means that this is just the disease in a dictionary or in a database that is linked with this. Yeah, and here we kind of say this is a change where we that is called a variant of uncertain significance. So every change in SCN2A, you can almost think starts as a variant of uncertain significance, and here we need more information. We need to kind of, ideally, we would like to know, is this change new in the child? Is this change a change that has been seen in healthy people? Yeah, then this would be kind of way less concerning. And at this point, just from the test that we have here, this would not be immediately clear. Yeah? There may have been other tests that have been done. There may have been features in the child that may be very suggestive, but here we can't make this statement yet. So next one. So again, this is this was a gene panel, probably. Yeah, you can see that there is there's this kind of big flag there that kind of labs often do to just kind of alert us to how this test is. And this is, I think, is this the same test? I mean, let me see. No, this is a different one. I think we've mainly seen previously there's an SCN2A change that is in a way uncertain. And again, it's the same. We don't have any more information about this change. It's never been seen in a different individual. We need to kind of, at this point, the best way to understand, is this disease causing or is this something that we're not concerned about? Yeah, is to kind of look at information from the parents. And if this, in case this is the same change we had seen previously, this was new in the child, and therefore it's probably explanatory. Well, I zoomed into this quite a bit. So this is now kind of a genetic test from my home country. This is in German. Yeah, and here we kind of, no, this is the C dot, and the P dot. And this is kind of classified as likely pathogenic. This change was new in the child. This is a change that has never been seen in anyone else before. And these changes typically are called likely pathogenic if they have certain additional criteria on what type of change this is. And typically, new changes in SCN2A in a child with epilepsy or developmental features, so changes that are de novo, not present in the parents, but present in the child. In the most cases, they are explanatory for a child's symptoms. Yeah, here I have kind of two tests at the end, which are quite interesting. So here we have a situation, again, where we have a certain change. And this is actually quite interesting, because this is actually a change that doesn't, at this point, is not kind of tested in the parents yet. Yeah? But they found a change, PM1872 threonine. So this is where this change has happened. And this is kind of marked as likely pathogenic. Yeah. And the lab can make this determination sometimes if there's a different change that has been known in a different patient. Yeah. And there's one patient who had a known new change at the same position where this wasn't a threonine but a different amino acid. And then under certain circumstances, this is kind of suggested to be disease causing, even though this particular change has never been seen in anyone. Yeah? And in this case, I would assume that this change would be new in the child, but this is the first test here, if I read this correctly, where the testing in the parent hasn't been done yet. Yeah, so this is a kind of a special circumstance. And sometimes there was a question earlier, does it matter where a change is? And the answer is, when it comes to gain of function, absolutely yes. Yeah, but there might be changes in the same positions where previous changes have been seen, and then there's a way for the lab to communicate this. And here is now the last change. Yeah, and this is now a kind of, let's say, the most complicated change that we've seen so far. I want to kind of finish with this just to wrap this all up. This is now a change. Let's kind of take this from the back. This is an SCN2A change, which is de novo. So a change that's new in the child, not present in mom or not present in dad, and is classified as likely pathogenic. And then we go back and see what this change is. And you can see this is one of these C dot P dots that are often a little bit difficult to understand. So it's not just a change of a single letter. So this is not I-1646 to a different letter, but here is some sort of additional language there. So ins GAT. And what has happened here, that this is actually an insertion of 3 base pairs. So this is, we kind of have seen previous reports that there can be frame shift variants. So you have your base pair, your kind of coding sequence of SCN3A that always happens in 3 different, or in 3 base pairs, coding for each amino acid. And if this is shifted by 1, if you take out 1 or if you add 1, this leads to a frame shift. If you insert or delete 2, this is also a frame shift. But if you insert or delete 3, it's a different story. You basically just have added 1 amino acid and everything else is the same. Yeah. These changes are often difficult to interpret, yeah? But we would typically think that this is also disease-causing in SCN2A if this is new in the child. Yeah, this is a change. As we've just seen from this protein structure, this is a channel, and you could imagine if you add a certain piece of protein there, this may kind of change what this protein looks like, and it's often resulting in a loss of function. So here we would kind of think of this change from the lab perspective, and this is likely pathogenic. You can see the C dot, P dot. The numbers are not that relevant. The main thing is that this is kind of an insertion, yeah. And this is a change where it's important to test this in the parents because we know that some of these changes might be benign, yeah. And here, this is a change that we think would be probably explanatory in the disease context. So I hope that this was kind of helpful going through some of these kind of changes. This language is very technical on purpose. So what is being kind of, what the language is kind of used for when we talk about C dot, P dot, and likely pathogenic or variant of uncertain significant or pathogenic, this is language that has been very, very standardized. So, and the idea was that 10, 15 years ago, there wasn't such a standardized language. So you could have a genetic test done in one laboratory and they would kind of think of this differently than the other lab. They use different language, yeah. And so this has all been kind of standardized now, that this is almost like a recipe that labs can follow, but they always get the same answer.
Thank you. Jessica, are you still with us? Do you want to ask your question now? I think
Sure, I can. Yeah, I wanted to ask if there's and I apologize if I missed this. I'm have a noisy child here. But is there a way to tell from the report if we have, if we're dealing with a gain of function or loss of function?
Jessica, could you type the variant that we're concerned about into the chat?
Yes, it was the one that you had said the verbiage was odd. It's the
I'm sorry. No, I wasn't. This wasn't. Was it this one here?
I was like, our report is broken. Yeah. C.584 greater than G, and then P.ASP195 GLY.
Let me have a quick look. This is the 195. So this is, we can, we have a database where we can look at what, if this change has been kind of measured at some point. At this point, it is not, we couldn't tell this necessarily from the genetic change itself. Yeah? So this is what we call a missense change. This is a spelling change that kind of just changes the ASP into a GLY, yeah. And these changes theoretically can be both, yeah. So those are changes that can result in a gain of function or loss of function. I'm just looking at our literature at the kind of clinical features of the patient that has been reported to have these features, which had infantile onset epilepsy, and we would think of this as a gain of function variant. Yeah. So this is from what we know from the clinical feature, is probably more likely to be a gain of function than a loss of function variant.
Thank you, that's helpful.
And for other families on the call, so with sometimes this is explicitly said on the report, but if you have a child who has a frameshift variant or a stop variant, those two variants, types of variants that we talked about earlier, the one with a star and the one that says FS in it, those are loss of function variants. Some variants have been tested in a lab to see how they affect the way the channel works, and some haven't. And this isn't always stated on the report.
And that line of questions, Omar asked, what does it mean if the report says intronic and doesn't have a P reference?
Great question. I'm going to share my screen again. I think a visual is good for this one. All right, let me get out of here. All right. So as we talked about, we have these two different sections of the gene. So we have the exon that provides the instructions for making the protein, the SCN2A protein. And then we have the intron, which is the genetic code between the exons and the introns. And so when our bodies are making the SCN2A protein, they go on what the exons tell them to do, and they remove this intron region. And so because of this, the intron isn't actually directly affecting the way that the protein is put together. And that's why it's not telling you a specific protein change, because the introns don't change the code of the actual protein. They can still cause disease if there's a change in an intron that changes the way the body puts together the exon. So sometimes these can be disease-causing variants, but they don't have a P dot because it doesn't change the code of the protein.
Thank you. Leah, did you want us to look at that variant?
Sure. Yeah, I think Stacy, I sent you two that didn't have reports that I just typed into the email. The one I just put one in chat. It's the GLN 1479 PRO.
So maybe one thing to kind of say is a variant interpretation on the fly is very difficult. Yeah, so this is something that so we can maybe pull a little bit of information to together there. What I typically like to avoid is just kind of make any determination there without having some of the information there. Yeah, and of course, I think one thing that's always important for us to say, especially in these kind of family meetings, not having the entire context. Please don't interpret this as medical information. And we do this basically in this informal family context. So let me try to kind of piece together what we can do about this variant, the 1479. Yeah. Do we know if this variant is de novo? Do we know whether this variant is new in the child and absent in the parents, or is this not known at this point?
I'm sorry, I don't know.
Okay, so this may be a common situation. This may just, and we can just kind of think about this hypothetically. This may just be a result from a gene panel that we did in clinic, and this comes back. So let's maybe walk this through hypothetically. So the first question is this a change that has been seen previously? Yeah, this is the main information that we can kind of go by right now, because we don't have any information about what the this change is. Yeah, I'm just kind of typing this in. So this is basically a change that I've seen previously, that we've seen previously in a child. At least the literature tells us that it's a change Q1479 in an individual who had neonatal onset seizures. So we would kind of, if this is not, this might be the same individual. Yeah, so this may not be kind of, always have to make sure that this is not, that we're not looking at the same individual over and over again. But if this is a recent test report, and there's another individual where this was a de novo change, we would think of this as a pathogenic variant. Do you have the other variant too, Leah? Would you mind just maybe typing this in quickly? Perfect. So here we have a bit more information. Stacy, do you want to kind of talk through this theoretically? So again, this is not basic, this is not a clinical interpretation that we're doing here. We're just kind of trying to apply these kind of rules in this kind of hypothetical context. So we just say, what is the information we would have here?
Right, and I think this is actually a good example of what we were just talking about. So this is an intronic variant. So one of those variants where you're not going to get a P dot because it's not directly incorporated into the protein. And the reason I know it's an intronic variant is because of that plus sign. Yeah. That's how the labs tell us that. So it's saying at this position, there should be a T and there's a C. So one of the major pieces of information that we think about when we're interpreting these variants, or one of the first things we think about, is this brand new in the child, or is it inherited from a healthy parent? So we can see here from the information that Leah kindly provided that this variant is brand new in the child. So that's a piece of evidence that we use to that leans us towards this variant potentially being disease causing. Also, the position in the intron matters in terms of whether it will affect the way that exons are put together or not. And we know that changes at +1 or +2 can sometimes, those are the positions that we're more concerned about when we're thinking about these types of splice site variants.
Yeah, so I think what we would kind of think about this is that this is a kind of de novo splice site variants. And when we think about how this would affect the protein, this would affect the protein in the same way as if this was a stop mutation. This is a kind of change where we would think that this affects how the SCN2A protein is being spliced together, is being stitched together. And this may result in the same consequence as if there was a stop change or if there was an entire deletion of this change. And this would be one way of interpreting this.
Like there's another one.
So in that case, it's likely, sorry, Jenny, just in that case, it's likely a loss of function.
Yeah, so in this case, this is what we call, so the stop mutations and frame shift variants and splice mutations, we kind of sum them together as protein truncating variants, and this is very likely a loss of function. No? I'm just reading that. I figured out how to read the chat while I'm kind of talking to everybody. So there's a question from Renata, kind of said, does every child with mutation in SCN2A have an epileptic and developmental encephalopathy? Yeah, and so when we think about what SCN2A related disorders can present with, so the features that children have, this can be different between children. Typically, the kind of three main groups are children who do have early onset epilepsy. Yeah? There are children who have mainly autism, and then there's a small group of children who have benign seizures in the first year of life. And I mean, this may be referring to one of the test reports that we had previously that list the change in the gene and then just kind of added just the disease name in the database. But this is just how labs sometimes do this. It doesn't say anything about what this change is or what a child has.
Yes, I can see where that would be really confusing if you got your report and didn't have a counselor to talk to.
And then there's one additional question about a change at position 604. Yeah. And I'm just kind of going through our database here, whether I find something at this position. I don't find anything there, and without, let's say, any further information, without kind of knowing whether this change is new in the child, or what features the child has, whether it's a change that's been found in healthy individuals, we would kind of call this finding this uncertain, and that we need more information to kind of understand whether this is disease causing or not.
We are about out of time. Do we have any last minute questions?
Yes, just one question there. What I mean, just to see if the approach that you use to get information is that, I mean, if I have a variant and there is someone that has the same variant, this is the kind of information that you're taking as a data. I mean, I have the question is, if someone else has the same variant, we should expect that they will have the same symptoms and effects. I mean, I'm not sure if you already have seen that kind of, your experience on that, if that has been happening or there is a lot of variation. I mean, it's not depending only with the variant, it's also other thing that could affect.
Yeah, so Omar, this is again a very, very good and very important question, that basically cuts right to the heart what our group is doing scientifically. And let's say the scientific term for what you're asking for is called a genotype-phenotype correlation. Yeah. Once we have a genetic change, what are the kind of changes that we know that, what are the kind of clinical symptoms that a child has, and are these symptoms the same in each and every child? So I have the following answer for you. So SCN2A is a gene where we see both. We see some changes. We know that every child has the same features or almost the same features. This is often some of the changes that cause early onset epilepsy. Some of the very common genetic changes in SCN2A have these features like the R853Q variant. But there are also other changes in SCN2A where we know that there's quite a wide range on how children can present. So this really depends on the specific change. Yeah? Many changes are very rare. So SCN2A is rare. And then of course, changes in SCN2A that are seen in more than one child are also rare. So we often have incomplete information, but typically from 2 to 3 children, we can get a sense on whether this is the same in everybody. Yeah?
And I have a further question. I mean, sorry. What you are expecting from the future? I mean, you think that maybe in 10 years we will have more data, and then we will have more information about every codification or variant? That is what you're expecting, or what else? I mean, just to see what will be our future, and maybe
yeah. So this is also again a very, very important question, and what I would like to basically limit this question for. So I don't want to talk so much about like the therapies, because I think this is part of a different kind of lecture that we would give. Right now, we only want to talk about the genetic testing. Genetic testing typically gets better with the more testing that's been done. And this happens in 2 different ways. This happens due to many healthy people doing genetic testing on like a research basis. So we know that right now there are hundreds of thousands of healthy people where we kind of can understand which changes in SCN2A they have. Yeah, in a few years, or maybe in 5 years, there will be 1 million of people. Yeah, so we would be better at understanding which variants are not disease causing, and this would kind of take care of some of the VOSs, these variants of uncertain significance that we're struggling with right now. Yeah, what we also know is that more and more children are getting tested, and this is one reason why we kind of really advocate for doing testing in epilepsy early, because we know once we find a change, and it's really important that this will affect our understanding of what the epilepsy or what the disease course is like. And with more children being tested on an ongoing basis, we would find more changes that happen in more than one individual. So we would expect that our kind of possibility of understanding what changes are will get better over time. You know? How well we're going to be, will we really understand each and every change? Probably not, but I think this will be significantly better in a few years from now.
Okay, we are.
And maybe then I think there was one last question about one change at the position 215. Yep. Let me just quickly look at our database here. And so this is not a change that I find in our database. So this is a change, this is very common for SCN2A. So some changes in SCN2A are what we call recurrent. So they happen in more than one individual. But what you will see with many of the SCN2A families, that they have changes that are very unique to a child and have never been seen before. Yeah, so this is very common. This is not unusual. You know, this happens quite frequently in an SCN2A, and this is one thing that is kind of very important to kind of know when we kind of think about what the clinical features are and how we can think about these changes.
Thank you so much, Dr. Helbig, Stacy. We appreciate your time tonight. And anybody, if you have any further questions, please email us at the foundation. And Diane, did you have something you want to say?
Yes, please. I had to notice that our variant encoding was mentioned. Could he maybe comment on it just very briefly?
Did you say you had seen it or not seen it? I'm sorry.
And tonight, and I have zero information on it. And our little guy has ketogenic hypoglycemia, severe autism, and recurrent seizures that they can't seem to get controlled. So it's inherited from mother.
Yeah. So
PN 604 that was up a little further.
Yeah. So the way I would think about this generally, so I think it is what we would typically do. I don't want to give medical kind of advice. I just want to kind of maybe take this as a bit of a kind of, let's say, general this discussion. So typically when we think that an SCN2A change is explanatory for a child's autism and epilepsy, then we would kind of typically expect this change to be new in the child. This can sometimes be inherited from parents who also have symptoms, but this is more the case for the benign SCN2A changes. So typically, most changes in SCN2A that are causing a severe disorder, they are new in the child and not present in the parents.
Correct.
So
It's not present in the
in this
parent.
case, we would say this would require some more kind of investigations, kind of saying, is this change that's also been seen in other healthy individuals? This information that I cannot really look up on the fly, but I think this is something where we would often advise families to kind of go back to your genetic specialist, go back to the person who kind of advised you about this test with exactly these questions. No?
I have no idea what it is.
Right.
Thank you.
So you've already talked to them, is that what you said? They don't know anymore?
Correct. We've had gone back 2.5 years after the original testing and to test for some mitochondrial differences because gaining weight, he has feeding issues and among other things, and they absolutely have no idea about anything about SCN2A.
Yeah, so it often it depends a little bit where you are located. It often, what many families do in this situation, that they kind of connect with other centers, or just kind of ask for other specialists, and this is often something that doctors and genetic counselors kind of work together as teams. And what I did maybe want to say as the last kind of a statement more generally is that there are some SCN2A changes that we don't understand. This is often a journey, and that it is always we'd like to get to an answer as fast as possible, but sometimes it's really difficult if there's a change that doesn't fall into any specific range, and there are some uncertain changes in SCN2A that at this point we do not understand.
Okay, thank you.
I think this is a good segue to just one last discussion of our team, if you would like to come and see us. So we have our team of child neurologists and epileptologists who all specialize in treating children who have genetic epilepsies or developmental differences. We have our team of genetic counselors. We have our team of physical and occupational therapists who all routinely work with children with genetic epilepsies. And if you would like to come see us, please email engine@chop.edu and we'd be happy to connect with you if you're interested in that. And I want to thank you all for your attention. It's been great meeting with you.
Thanks so much.
Thank you so much.
Thank you.
Thanks so much.
Bye.
Thank you.
Thank
Bye,
you.
all for you.
Thank you.
And thank you for the
Thank you.
coordination of this. Thank you.
Thanks.
Bye.
Have a good night. Thanks, Ingo. Thank you, Stacy.